What if some of the most powerful tools in the fight against cancer weren't brand new billion-dollar drugs, but were actually hiding in plain sight, already sitting in our medicine cabinets? Today, we're diving into the fascinating world of drug repurposing, looking at how common medicines for things like parasites and cholesterol are getting a second look as cancer fighters. And it all starts with a very, very strange connection. Okay, so take a look. On the left, you've got a parasitic roundworm. On the right, a complex cancer tumor. They seem about as different as you can get, right? But what if I told you that a drug designed to kill that parasite could also do some serious damage to that tumor? Well, that single idea is at the heart of a scientific strategy that's picking up some serious steam. So yeah, scientists are now looking at our existing drug supply as a kind of unexpected arsenal in the war on cancer. The whole approach has a name. It's called drug repurposing. And trust me, there are some really good reasons why researchers are getting so excited about it. So, what exactly is drug repurposing? Well, think about it like this. Creating a new drug from scratch can take a decade and literally billions of dollars. It's a huge gamble. So the idea here is, why reinvent the wheel? Let's investigate the drugs we already have. We know they're generally safe, they're often really inexpensive, and they're available right now. It's a massive scientific shortcut, and it's a pretty brilliant one. And when you look at the candidates in this repurposed toolkit, it's honestly kind of mind-blowing. We're not talking about obscure chemicals. We're talking about common antibiotics, diabetes drugs like metformin, anti-parasite meds, and even statins. Yeah, the same ones millions of people take for high cholesterol. The potential has been hiding in plain sight this whole time. All right, so here's the million-dollar question. How? How on earth could a drug for cholesterol possibly fight cancer? Well, it turns out that many of these drugs are like a Swiss Army knife. They launch a multi-pronged attack, hitting cancer cells in ways that original designers never, ever imagined. Let's take that dewormer, mebendazole, as a perfect example. This diagram shows its 5-point attack strategy. See, it's not just doing one thing. It goes after the cancer cell's internal structure, kind of like collapsing its skeleton. It also chokes off the tumor's blood supply. Then it blocks key growth signals. And on top of all that, it actually helps our own immune system recognize and attack the cancer. It is a full-on, multifaceted assault from one single unexpected drug. To get your head around another one of these really clever mechanisms, think of it this way. You're not just flipping the main power switch off. You're also taking out the emergency backup generator at the same time. And this slide shows that exact strategy in action. Some cancer cells are what you'd call metabolically flexible. A drug like the antibiotic doxycycline can deliver hit 1, shutting down their main energy factory. But the cancer cell just switches over to a backup fuel source. So that's where hit 2 comes in, blocking that backup pathway. With both its power sources cut, the cancer cell basically just starves. So with these clever attack plans, it's probably no surprise that the results from preclinical studies, the research done in labs and on animal models, have been incredibly promising. Check out this number, 129%. In a mouse model for a really deadly type of brain tumor, treating them with the dewormer mebendazole led to a 129% increase in median survival. That's more than doubling their lifespan, an absolutely staggering result. And it is not a one-off. In a totally different study, the common antibiotic doxycycline caused a 70% reduction in the total tumor burden in a mouse model of breast cancer. I mean, these are the kinds of dramatic preclinical results that get scientists really, really excited. And the promise isn't just in animal models. One study that looked back at data from patients with specific types of blood cancer found that those who happened to be taking statins had a 61% lower risk of dying from their cancer compared to those who weren't. So after seeing all this, you're probably thinking the obvious question. If the results are this good and the potential is so great, what's the holdup? Why aren't these drugs being used in cancer clinics everywhere? Well, that leap from the lab to the clinic is a huge one, and it is full of challenges. And one of the single biggest hurdles is something we can call the dose dilemma. It's a fundamental problem that really comes down to concentration and safety. Here's the issue in a nutshell. In the lab, in a petri dish, you can just flood cancer cells with a really high concentration of a drug. But you can't do that to a person, right? You're limited to a dose that's safe and tolerable. And very often, that safe dose results in a much, much, much lower concentration of the drug in your actual bloodstream.
This chart shows the problem perfectly. Let's take the antiparasitic drug ivermectin. This huge bar on the left, that's the concentration you need to kill cancer cells in the lab. And these tiny little bars on the right, that's the maximum concentration you can safely achieve in a patient's blood. This enormous gap is the dose dilemma, and it's a huge reason why so many promising lab results just fail when they get to human trials.
It all really comes down to one of the most crucial lessons in all of medical science. The human body is just infinitely more complex than a petri dish. What works in the lab simply doesn't always work in a person.
So what does the future hold? How do we take all this amazing promise and responsibly bridge that huge gap between the laboratory and the clinic? Well, the path forward requires a lot of patience and really rigorous science. It means we need carefully designed human clinical trials to find the right dose. It means investigating these drugs in smart combinations, and of course, making sure safety is always the #1 priority. We also have to solve tough problems like bioavailability, which is just a fancy way of asking how much of the drug actually gets absorbed by the body to do its job. It's a slow, methodical process. And this brings us to the most important takeaway of all. While this research is incredibly exciting, it is not a set of instructions. It's not a do-it-yourself guide. As experts like Dr. Simcock constantly emphasize, any questions about cancer treatment must be discussed with your healthcare team. Self-experimenting with these drugs is incredibly dangerous. So, after looking at the incredible promise on one side and the sobering reality on the other, what's the key takeaway here? It's this: the search for new uses for old drugs is one of the most vibrant and vital frontiers in cancer research today. It offers a path to treatments that could be faster, cheaper, and safer. But, and this is a big but, we have to remember that promise is not proof. The scientific process has to be followed to turn these exciting clues into proven therapies. And that really leaves us with a final, fascinating thought. This research proves that sometimes revolutionary ideas can come from the most unexpected places. It just makes you wonder, what other secrets are hiding in our everyday medicine cabinets, just waiting to be discovered?